So our next speaker is going to be Dr. David Hyde. He is a professor of biological sciences here at Notre Dame. He is also the Reverend Howard J. Kenna Memorial Director of the Center for Zebrafish Research. And so his title is up here, and there's nobody got to say.
Yeah, probably a little bit. Can you get rid of these? It's all or none? Okay. Okay, well, thank you for coming. I'm going to be talking to you a little bit about a specific application of stem cells this afternoon, and in particular, the ability for stem cells to regenerate nervous tissue, and regenerate not just in a morphological sense, but also in a functional sense. So there's a variety of different diseases that exist within the human population. Several are listed right here, which lead to the degeneration or loss of specific cell types in different types of tissues, some of them being neural tissue, such as macular degeneration, spinal cord injuries, Alzheimer's disease, and other types of diseases that are not affecting neural tissue. In all these cases, the therapies that exist for these diseases are basically sort of stopgap measures in which we try to, or the medical field tries to keep the tissue from becoming further damaged and retain as much function as possible. But there are not good examples or therapies available right now to try to regenerate the damaged tissue itself. So in the center here, we've got a picture of the Golden Dome on a bright sunny day, not like today though. And then we have 4 different retinal diseases that afflict the human population: retinitis pigmentosa, macular degeneration, which is the most common form of blindness in the elderly, diabetic retinopathy, and glaucoma. And what you have is an image of what the dome would look like in each of these 4 diseases. And again, these are degenerative diseases. So as you begin to lose your vision, the best we can do at this point in time is to sort of slow down that vision loss. We cannot recover any functional vision. So as you start to lose your peripheral vision in retinitis pigmentosa, there are treatments that will slow it, but it will not recover that peripheral vision that has been lost. So the hope or the goal, one of the goals of stem cell therapy is to be able to regenerate damaged tissues. And as Rebecca showed earlier today, we can take pluripotent stem cells of a variety of different sources and hopefully be able to transplant them back into individuals and be able to regenerate tissues that have been damaged due to a variety of different diseases. But just as importantly, these stem cells will lead to, if we work with them and study them, we'll have a better understanding about a variety of different processes that are required for normal development and birth defects, and understand the underlying pathologies behind these diseases also. So there's a lot to be gained from it besides just from a regenerative standpoint. As Rebecca mentioned, embryonic stem cells, which are a highly controversial subject in the case of humans, are derived from cells in this inner cell mass. And people have tried to use embryonic stem cells, primarily using mouse embryonic stem cells, to regenerate nervous tissue. And in these cases, in which you have mouse models for human diseases such as retinitis pigmentosa, when these embryonic stem cells are injected into a damaged mouse retina, they begin to proliferate in an uncontrolled manner, and they start to differentiate into cells in an uncontrolled manner. So that not only do you regenerate neurons, but you regenerate muscle tissue, neurons, and a variety of other different cell types. So this leads to a non-productive regenerative event, because in these human diseases, 1 or 2 specific types of cells are lost, and those should be the only types of cells we try to regenerate, not a plethora of different cell types. And so this resulting uncontrolled proliferation and differentiation failed to lead to any sort of functional regeneration or recovery of vision in these mouse models. So besides the variety of different ethical and moral and legal concerns about using embryonic stem cells for regenerative medicine, there's also a scientific concern associated with using them. As of this time, they have not been used successfully for any sort of regenerative biology in the nervous system. Rebecca mentioned iPS cells, or induced pluripotent stem cells, and these are cells that were first demonstrated by Yamanaka's lab, in which you can take a somatic cell, either introduce 4 specific genes, or as it's been modified subsequently, RNAs, proteins, small molecules to activate those genes or their protein products, and convert that cell, that somatic cell, back to an embryonic stem cell-like fate or identity. And so these cells hold a lot of promise because not only can we generate cells from an individual person that could be used as a potential therapy for that individual, but also these iPS cells can be used as disease-specific models. So we could take individuals that suffer from a particular type of disease, such as Parkinson's disease. We can generate iPS cells from their fibroblasts, their somatic cells, take those iPS cells, convert them into neurons, and then look to see what the defects are within this individual who is suffering from Parkinson's disease. However, I think what's been shown very recently is that there are potential limitations to this. So a group down at the Salk Institute recently took somatic cells from patients who suffer from schizophrenia, converted those somatic cells into iPS cells, then took those iPS cells, converted them back into neurons, and they've identified now that these schizophrenic neurons that were generated in culture possess reduced neuronal connectivity. They have significant changes in gene expression relative to normal neurons. And so we're left with a sort of a question when we're looking at a complex tissue such as the brain, and we're looking at a disease of the central nervous system, the brain itself, such as schizophrenia. Do these simple sorts of defects that we observed in this iPS cell model, are they really telling us a whole lot about the disease? Are they going to give us a lot greater insight into how we can maybe generate therapies for this disease? Or is it just simply going to be sort of a long path to try to get at some sort of therapy? So while there's a lot of potential, I think we need to be careful about the sorts of diseases that we tackle with these cells. Now, there's a variety of potential difficulties also associated with iPS cells. So they've been found occasionally to be limited in their pluripotency. So they may not be able to differentiate in every cell type within the individual. And in some cases, when these cells are injected back into the donor, they have been rejected. And this is using mouse models again. So what's been argued as one of the advantages of the graft rejection issue isn't as clear-cut as what it appears to be. When you introduce these iPS cells into an individual, you also form teratomas, which is uncontrolled growth of both ectodermal, mesodermal, and endodermal tissue, much like if you introduce embryonic stem cells into an individual. These iPS cells have been shown to acquire a large number of mutations that were not present in the original somatic cell, so the process of reprogramming these cells goes through a mutational type of event which is uncontrolled at this point in time. And many of these types of mutations are tumorigenic. These are not due specifically to the 4 genes that you're introducing into those cells, but it's due to other accumulated mutations that are occurring, and nobody understands why this is occurring. And then finally, while these iPS cells appear to be reprogrammed back to an embryonic stem cell-like fate, they still contain epigenetic markers that were found in the original somatic cell. So they have not been converted totally back to an embryonic stem cell-like fate. They still retain some sort of identity to the somatic cell they were derived from. Now, I say these are potential difficulties. I don't think this is going to greatly limit or reduce the potential for using iPS cells, but it does raise some issues that need to be resolved scientifically before they can probably reach their full potential. Again, people have tried to use iPS cells to correct neuronal difficulties, and using the exact same mouse models of retinitis pigmentosa and cone cell degeneration or macular degeneration, again, these iPS cells fail to regenerate functional neurons in this model due to the fact that, again, there's this uncontrolled proliferation that's going on, their failure to differentiate into only the desired types of neurons that are missing. And then an additional problem which has recently been identified is that injection of these iPS cells actually fail to actually integrate into the retina. So that there's physical barriers, limiting membranes that prevent this integration. But while this is a potential difficulty, it's also been resolved within the last year by using specific types of enzymes such as MMPs, in particular MMP2 or metallic proteinase 2, we can get these cells to integrate into the retina. A third type of cell, which Rebecca sort of hinted at a little bit, is progenitor cells. So as these stem cells divide, they produce not only themselves again through the self-renewal process, but they also produce progenitor cells. And progenitor cells have been shown to be much more effective at dealing with these neuronal defects. So if we take a specific type of progenitor cell, rod photoreceptor cell precursor, and this is designated by this NRL+, it's expressing a specific protein. This cell has stopped going through cell division, so it's post-mitotic. It is now committed to ultimately becoming a rod photoreceptor cell. We can take those cells, inject them into a variety of different mouse mutants in which the rod photoreceptor cells have died, and these progenitor cells can be integrated functionally into the retina at a very low level, but it does occur. And what you actually see is, from a functional standpoint, it increases their threshold to light by an order of magnitude of 1 log, which is 1 log order less than the wild-type retina, but 1 log order better than the blind mouse retina. So what this suggests then is possibly the use of iPS cells, not in using iPS cells themselves, but taking the iPS cells, growing them in culture, and then pushing them down a particular pathway of differentiation so that they become progenitor cells, they exit the cell cycle, and then we can introduce those back into the damaged nervous system. So what this also suggests is that pluripotency may not be all that it's cracked up to be. That maybe unipotency or limited potency may be more advantage in regenerating a variety of different tissues. So that if you know you're trying to regenerate beta cells in a pancreas, the fact that the cells you're introducing can't become neurons or can't become heart tissue is not really a problem at all, as long as those cells can efficiently and effectively regenerate beta cells within the pancreas. So the last type of cell that Rebecca talked about were adult stem cells. And as she mentioned, these exist in most human tissues, but they exist, it appears, in a relatively quiescent sort of state. They might be proliferating at a low rate, they might be actually non-proliferating, but still have the ability to re-enter the cell cycle. And in some organisms, damaging the tissue is sufficient to reactivate these stem cells. So, to give you an idea of how I sort of look at this problem, is we have a tissue right here, which is simplified as a variety of different cell types, each cell type having a particular shape and a particular color. If we damage that tissue, we lose one of those particular elements and then add back a stem cell which is pluripotent, those cells will integrate randomly within that tissue, and then they will randomly identify particular differentiation states that they're going to become, and you generate this sort of pattern, which is very different than what you have up here. And so this is clearly not going to be a normal pattern, a normal tissue. It's probably not going to be functional. And this is what we see right now when you introduce embryonic stem cells or iPS cells into a damaged tissue. In some tissues, in some organisms, what happens is this damaged state induces a particular type of cell, such as these red triangles, to de-differentiate or become an earlier fate or re-enter the cell cycle if they're quiescent. And those cells then have the capacity to proliferate. Now all of a sudden these cells are already integrated within the tissue and are potentially in a better position to identify what particular type of cells are missing. They can proliferate to become that particular type of cell and then differentiate to recover this normal pattern that you originally had. And now you have an opportunity for this to be functional also. So my lab utilizes zebrafish, as does Rebecca's, for our research. And we utilize zebrafish for a variety of different reasons. They're small in size, which means we can keep hundreds of thousands of these fish right here on Notre Dame's campus. They generate a large number of progeny from a single mating. So we can take one male, one female, and a single mating can produce upwards of 200 progeny, which allows us to be able to do a variety of different types of genetic things that are more difficult to do in other sorts of systems, such as mice. We can isolate a variety of different mutants. We can isolate mutants that mimic human diseases. We can isolate mutants that have defects in specific genes that mimic human diseases. The genome is largely sequenced, so we can target whatever gene we want. And finally, they have the capacity to spontaneously regenerate a wide array of tissues. So you can do things such as cut off their fin, and the fin grows back. Okay, fairly simple experiment. You can cut away a third of their heart, and the heart grows back without any sort of scar tissue. A remarkable feat. If we could do that in the human population, can you imagine what that would do to medicine? It would revolutionize it. And as I'm going to show you, we can regenerate neuronal tissue, functional neuronal tissue. And so the goal here of the work going on in my lab is to understand this process in this system, in zebrafish, where we can genetically and molecularly dissect things out. Often, people, friends of mine, will say, well, if you're interested in trying to cure blindness in humans, why don't you do this with humans? And as I hope all of you appreciate, we cannot ask for human volunteers to come forward, mutate their sons, and then mate them to all these different daughters that exist within the population. We just can't do this. But we can with fish. Nobody cares what we do with the fish. So the fish retina is very similar to the human retina. Okay, so is the adult fish retina right here. You can see it has these different layers of darkly staining cells. These represent the cell bodies. So we have a ganglion cell layer, we have an inner nuclear layer, and we have an outer nuclear layer. This is the same sort of arrangement you see in the human retina. This schematic shows the variety of different cell types within that retina. It's a fairly simple neuronal system containing 7 different types of neurons. Okay? One neuronal cell, which is shown here in blue, which is called the Mueller glial cell. All of those cells are present in the human retina. This Mueller glial cell turns out to be an adult stem cell, which resides within this tissue. Now, this adult stem cell, this Mueller glial cell, serves particular functions within the retina. It's there to make sure that we are not producing toxins, that we're producing the right type of growth factors to keep the neurons happy. It's there to actually function to channel the light which is coming through the lens, through all these different nuclear layers, out here to the outer nuclear layer, where the rod and cone photoreceptors are, so we can see an undistorted image of whatever we're looking at. So they normally carry out these processes, but in the fish, it carries out one additional process. And in the zebrafish, the fish grows throughout its entire life. So as it grows throughout its life, it has to make new cells for all of the tissues, including the central nervous system, of which the retina is part of. And so what I have here is an image, which is very dimly lit here, showing proliferating cells. And here is a cell which is proliferating in the internuclear layer. And under normal circumstances, this cell, this internuclear stem cell, as it was called back in 2003, divides asymmetrically to regenerate itself or renew itself, and it produces progenitor cells. And these progenitor cells then migrate from the internuclear layer to the outer nuclear layer, where we call them rod precursor cells, because at that point they are committed to becoming a rod photoreceptor cell. They will not become a cone photoreceptor cell. They will not become a Mueller glial cell. They will become only rods, as shown in this lineage right here. So this is under the normal circumstance, and it happens throughout the lifetime of the fish. In situations when this fish retina is damaged, such as when we put them in extremely bright light, what happens is this retina goes from looking like this to within 4 days looking like this. And the two things that happen are, first, the obvious thing is this region becomes much smaller. And this is where the outer segments, or the part of the rods and cones that detect light, are located, and that part is lost. The reason that part is lost is because the rod and the cone photoreceptor cells themselves are lost or die. And that can be seen because this region of blue dots here, which represent the nuclei of the rods and cones, go from several nuclei thick to basically one nucleus thick. And we can count this and quantify this a variety of different ways. But what happens is if we were to put this fish back into standard light, take it out of the very bright light that caused the damage, and wait 30 days, actually even less than that, maybe about 14 days, we can actually regenerate this retina. So these outer segments have returned, and this outer nuclear layer, again, is several nuclei thick. So we get a morphological, structural regeneration of the retina in a very rapid time. This happens spontaneously, and how it happens is because these Mueller glial cells, shown here in a transgenic line that we generated in the lab, which expresses green fluorescent protein in just the Mueller glial cells. These cells, 16 hours into the light damage, the retina would look like this. The Mueller glial cells are still present. They're still expressing GFP. By 31 hours, they now start expressing a different protein, pCNA, a protein which is expressed in cells that are trying to divide. So these Mueller glia are starting to divide. They then produce cells that are now red, expressing pCNA, but no longer expressing GFP. These cells represent the neuronal progenitor cells. So that Mueller glial cell has divided, regenerated itself, produced progenitor cells, and these progenitor cells are now continuing to divide or amplify, as Rebecca mentioned this morning, and they begin to migrate to the outer nuclear layer where they will differentiate in the rods and cones. Question, sure.
Quick question. If you look at before and after slides
Yeah.
at the top, they don't look exactly the same.
Because this is not the exact same genetic background.
Got it.
Okay,
Thanks.
good question. That way I know people are awake. Seriously. Okay, so this is what happens when we damage or kill off rod photoreceptor cells. What happens if we kill off other types of neurons? And so we can take a particular drug called wabane, which is a neurotoxin, inject it into the eye at very low concentrations, and this is our control. 3 days after the injection, what happens is this ganglion cell layer and this internuclear layer have now vanished. And the outer nuclear layer, where the rods and the cones reside, remains intact. And over the course of 60 days, it regenerates. So it becomes the converse of the light damage model, where we kill off rods and cones. We kill off all the other types of neurons. And what happens is, again, this is our transgenic line expressing GFP in the Mueller glial cells. They begin to start to express pCNA as they divide. They produce progenitor cells, and they start to migrate to wherever the damage is. So there's a morphological regeneration that occurs, a structural, a cellular regeneration that occurs. Is there a functional regeneration? Because keep in mind that these new neurons are now being born within a neuronal network in which it has to find the proper synaptic connections. A very complex issue for these cells to do. So this represents a behavioral test that we did with Lei Li here at the University of Notre Dame. We put this fish in a circular tank. It cannot see across this post, and so the fish swims basically just in circles. We can then put on the outside of the tank this black stripe, and we can rotate it in whatever direction we want at will. The fish, while they are remarkably good at regenerating, they are not very bright, okay? They think that this black stripe is Shamu the killer whale or something, and will do everything they can to avoid it. So this fish, 2 weeks earlier, just swam in circles around the tank and totally ignored the black stripe. Now, 2 weeks later, after all that morphological regeneration has occurred, you will see as we change the direction of the stripe, so that the fish sees it, it will change its direction. And on 10 out of 10 encounters with that black stripe, it successfully avoided it. Even when we tried to trick it by changing how long it takes before we switch the direction of the stripe. So clearly, this fish has been able to reconstitute visual behavior as best as we can assay. We've also been able to do electrophysiological tests, and those allow the fish to look like it's also functionally regenerated. So when we talk about regeneration, the question that comes up is that basically, is this regeneration just recapitulating development? Are we causing these stem cells to basically go through development again? And so this is what the retina looks like very early in development, 30 hours after fertilization. It's a mass of a single epithelial layer of cells, undifferentiated, and within about 3 days, we have generated different types of neurons, and they are separated into these different layers. And then this persists until adulthood. When we look at the light damage model, which is what happens here where we lose rod and cone photoreceptor cells, this is remarkably similar to what you would see in development, where the rods are the last type of neuron that are born. So in this sort of situation, the progenitor cells that are being produced from the Mueller glia are seeing an environment which is very similar to what they would see in development. In the Wabane damaged retina, it's a complete opposite, because now the only neurons that are left in this damaged retina are the rods and cones, which are the last neurons to be born. And now we're asking it to create all the earlier developing neurons and wire properly into cells that it wouldn't normally be trying to connect with because they aren't there under normal developmental circumstances. But in both cases, we get normal regeneration of the tissue and functional regeneration of the tissue in that behavioral assay. So if it is not development that seems to be happening, is there some other process that seems to be being mimicked here in the regenerating zebrafish retina? And what I would argue is that the process that we're seeing is something which is very similar to reactive gliosis that we see in the damaged human retina. So if there were any ophthalmologists that were in this audience, which I don't think nobody has a background in ophthalmology? No? Okay. So whenever you damage the human retina, what happens quite often is the Mueller glial cells, okay, after this damage, begin to swell up. They express specific proteins such as GFAP, an intermediate filament protein, okay, glial fibrillary acidic protein. After expressing these proteins and starting to swell up, the Mueller glial cells start to divide. This is exactly what we see happening in our damaged zebrafish retina. The same sort of molecules are being expressed, the same sort of swelling of the Mueller glia is occurring, and the proliferation is occurring. However, what happens is in zebrafish, the Mueller glia divides once, maybe twice, to produce progenitor cells, and the progenitor cells then migrate and differentiate into the missing neurons. In the mammalian retina, we fail to terminate the Mueller glia proliferation. They continue to divide, they start to migrate and create a glial scar. And so we think that there are similarities that are occurring initially here between these two processes, one occurring in humans, one occurring in fish, but there's a difference in the outcome that occurs down here where we have a glial scar, we have a functional regeneration occurring over here. We don't know, and I think it's impossible to tell where these two pathways diverge genetically. It could be happening very early in the process, but is not manifested until later in the process, until we can see what's happening, what's proliferating. And so we've taken the tact of asking what is happening during normal regeneration as a retina is damaged, producing a signal to the Mueller glia to begin dividing. This Mueller glial cell divides to produce a progenitor cell. The progenitor cells then continue to divide or amplify a number, and then they begin to migrate to the proper damaged layer of the retina and differentiate into the missing neurons. So we've taken a variety of different tacks. We've asked what sort of molecules initiate this proliferation response. I'm going to move through this rather quickly now, okay? We generated a model in which we took a fish, we damaged it in bright light to the point in which we know that the signals, whatever signals there might be to initiate proliferation are occurring. And we take the eye from this fish, grind it up, and inject it into an undamaged fish eye. So if there are molecular signals that are causing this regeneration response, the molecular signals from this damaged eye should now be present in this undamaged eye and trying to cause regeneration to some extent in an environment where there is no damage. And if that's the case, can we see regeneration? We can see a regeneration response. We can isolate the protein changes that occur between the damaged retinal lysate, and we've identified one particular protein, protein spot 90 right here in purple, which corresponds to tumor necrosis factor alpha. And if we look at that protein down here in the damaged retina, TNF alpha in green increases in expression 16 hours into the light damage, the point in which cell death is maximally occurring. And by 35 hours, when the Mueller glia start to proliferate, we start to see it down here in the Mueller glial cells. We can do RNA experiments to see what cells are expressing this TNF alpha. And at 16 hours, it's the rods and cones that are expressing TNF alpha. And in fact, we can look at which rods and cones are dying, and it's actually the dying rods and cones that are expressing TNF alpha. So the right type of cell is expressing this molecule at the right time to be signaling, hey, we're dying, you better start regenerating. We then developed a technique in which we used Morpholinos, which are very similar to siRNAs or microRNAs, in that they bind to a particular mRNA sequence and block the expression or translation of a specific protein. When we do that, we can block the expression of TNF alpha, as shown here, relative to the controls. And when that happens, we block the proliferation that's occurring of the Mueller glial cells in the internuclear layer relative to the controls. And we can quantify that. So the green bars represent where we block TNF alpha expression, the blue and the red are the controls. So it appears that TNF alpha is a molecule that's required to initiate this proliferation response. I identified other molecules shown here, ASH-like 1A, STAT3, SOX3A, that are also involved in this response. But the question was, could we identify molecules that are expressed later in this regeneration? And so we did a gene microarray experiment looking at changes in gene expression. We identified over 1,700 genes that were increasing in expression during this time course. And we looked at one particular one, PAX6. There's two PAX6 genes, a PAX6A and a PAX6B. These genes, while they're expressed initially in a variety of differentiated neurons, shown here in green, in the undamaged retina, once we damage that retina, at 51 hours into the damage, we see PAX6 expression coming up in these progenitor cells. At 68, it's still there, and at 96 hours, it's still there. So again, we used our Morpholino technique, block PAX6A and block PAX6B expression. And what we found was that when we block PAX6B expression, we can allow that Mueller glial cell to divide one time to produce a progenitor cell, and that progenitor cell cannot divide any further. If we knock down PAX6A expression, that progenitor cell can divide a couple of times and then stops, but does not reach its full proliferation maximum number. And so clearly PAX6B and PAX6A are required downstream of initiating this response. So why is this important? Because in the other model people commonly use to study stem cells in the retina, the mouse, when you damage the mouse retina, the Mueller glial cells at a low rate will start to divide. And when they divide one time to produce a progenitor cell, and then it stops. It's exactly what we see when we knock down PAX6B. That progenitor cell then differentiates into whatever type of neuron it wants to and ultimately dies. So what we're trying to do at this point is utilize zebrafish to try to tweak out what are these different processes that are controlling its spontaneous regeneration. We've identified several that initiate the regeneration response, and we've identified molecules that when we knock them out, basically mimic what happens when we look in the mammalian retina. So we're now starting to form collaborations to look at what's happening to the expression of these genes in the damaged mouse retina. And so I think that there's a variety of different human degenerative diseases that exist that require a regenerative type of therapy. And this is the only way we're ultimately going to be able to reduce the suffering from the individuals that have these diseases. Human embryonic stem cell research, which is based largely on, I believe, the historical mouse embryonic stem cell research that has been going on for several decades now, has yet to produce any tangible regenerative responses. So I'm not sure that we are either vastly underestimating how to utilize those cells in a regenerative capacity, or they lack the ability to be that specific for a regeneration response. So adult stem cell biology, while it does have a variety of different problems, such as they're few in number, they divide very slowly in culture, and so their ability to isolate them, grow them, and put them back into a tissue to regenerate tissue looks like it's probably not going to be a very effective means, has the ability to spontaneously, within the organism itself, regenerate tissue. And that might be where this type of regenerative therapy, at least with adult stem cell, lies right now. Utilizing something like zebrafish to sort of point us in the right directions allows us to then direct our efforts, as opposed to saying, we want to make a mouse retina regenerate. What are we going to do? And just randomly trying a variety of different things and trying new things over and over again until we try to hit upon things. It should be mentioned that Yamanaka's group, when they identified this iPS stem cell technology, they identified about 30 different genes that they thought were important. They tried each gene independently, failed to get an iPS cell, started to do pairwise combinations, 3 genes at a time, ultimately 4 genes at a time before they actually got this to work. So trying to sort of shoot in the dark is not the most productive way of going. And so we think utilizing zebrafish should help. And so the people in the lab that have been doing this work, several postdocs, graduate students and undergraduates have been working on this project. The people whose names are bold are people who provided data for this talk. We have been funded by the National Institutes of Health, Fight for Sight Postdoctoral Fellowship, the Johnson & Johnson Focused Giving Program, and the Center for Zebrafish Research at Notre Dame. And I'd be happy to answer any questions.